Thank you very much for this. This is the first time I've presented this to kind of a general audience. So if I look surprised when a slide comes up, it probably is because I'm surprised. So what I'm really going to talk to you about today is using code, machine learning, to understand our code. And really it's the thing that Shankar's been talking about, is driven by this code. And we are one of the most complex machines ever built. So not surprisingly, it's very complex. But just to give you some perspective on why it's also complex is its inaccessibility. Because where I work, not up the hill in the JR, but actually where my brain is most of the time, is deep, deep inside the body. So this is down to what you could do with a magnifying glass. This is like a rubbish microscope, and then this is a professional microscope. And that's the front door of where I work. Where I really work is in here, the nucleus. This is basically a CPU preloaded with code that drove all of the beautiful things that Shankar showed you, and every function in your body as an adult. So it's small, but it's incredibly complex. So you've got 3 billion letters in that tiny little space. There's about 2 meters of it if you stretch it out, which is considerably taller than me. So it is incredibly complex, and that's one of the problems that we need AI for, or machine learning. When we sequenced the human genome, we knew about one thing in it, and that's genes, things that make proteins. So we then had a look at, that's the whole genome. The little red slice is the bit we expected to be functional. So there's a huge amount of other stuff that somebody, not very helpfully, said was junk. It is not junk. It's incredibly important and incredibly beautiful, actually. So this is your DNA, and this is how you read DNA. So that's the classic double spiral you've seen in everything. So it's actually just not naked DNA. It's actually packaged in a very ordered way. And so these nucleosomes, which are collections of proteins, basically act as a packing. But they also encode this fantastically layered organizational, basically, computer. And there you get back out to the cell again. So what is it actually doing? The way you read your DNA code is to turn it into another molecule called transcription. So here, if you've got your DNA, this little motor is this is a very simplistic version of it. But what it actually does is it whizzes along and basically copies it into another molecule called RNA. It's got the same 4-letter base code that the DNA is, but it's actually a different molecule. And what you can see is it's just unwinding it, pulling in the molecules from the surrounding nucleus, and actually just copying the code. And it does this incredibly quickly, which I should show in a minute. Go. Zoom. So what is it actually transcribing? These are the genes that I was talking about. So what you have in this piece of DNA is things called exons and introns. Exons are the bit that actually hold the information. And for some reason, we have breaks in it called introns, and we're not 100% sure why. It gets copied into another molecule that has exactly the same structure. At that point, it then gets rid of the introns. And what that does then is taken out of the nucleus into a part of the cell where it's actually then used to make the proteins we're made of, essentially. And this is actually hemoglobin. This is the thing why your blood's red. So is the protein code the reason why we need AI? Not really. You can actually decode it using the back end of an old textbook and a piece of paper. I can't. It's very simple. It's read in a strictly linear order, and it's been the same for about 3.5 billion years. Also, if you look at the mental complexity, this little thing on the left is a worm called C. elegans. It's got about 1,000 cells, not very many different types of cells. We're on the right, we've got trillions of cells. We have no idea how many types of cells are in us. And we have pretty much the same number of genes. So where does all this complexity come from? It comes from the junk. So what's in the junk? They're regulatory switches. Because what the complexity comes from is not the number of genes, but how you can turn them off and on in different combinations to arouse complexity. And they tend to be called enhancers. So as Shankar showed you, you go from a very simple cell, and you actually take that one genome, it's got the same code and CPU in every single one of those cells, and actually makes all of these different cells and maintains and functions all of those cells. So how do they work? This is the anatomy of one of those enhancer sequences. So the thing on the right is a protein called a transcription factor. It's actually also produced just the way I showed you. So there's a chicken and egg here. You have transcription factors running the process to make proteins, and they're proteins anyway. You get them from your mother if that's the question you're going to have. They bind DNA, but they just don't bind any old DNA. They bind specific words in that DNA, and you have A, G, T, and C in your genome. So this is actually a real enhancer, and what I've done is lined up the genomes of other species. And the trick here is if it's functionally important, it will stay the same. So the black bits that are actually the same in all these different species, or the red bars, are transcription factor binding sites. And the reason why each one doesn't get bound is because they have to cooperate to make this functional element. And that's a fundamental principle of how this works. About 10% of your genes are transcription factors. So there's a massive complexity of different interchanging combinations that actually drive your different cell systems. So you can think of it that every cell type in your body uses a completely different code. That's why we need machine learning. Enhancers in genes. So the little bar is a gene. These switches they're really annoying actually to work with. They pop up right beside the gene. They can be 1 million base pairs away from the gene. You can have other genes in between them that can be buried in the body of other genes. You can have many of them all driving the same gene. And then you turn it into another cell type, and then they all move around again. And another gene turns on. That is part of the actual complexity of this. And it's very hard to predict which switches are driving which genes. So how do we find the switches? This is an analogy which is probably apt to consider the amount of sort of art going on in this. It's etching the genome. So if you remember how you used to etch glass, they used to cover it in a sort of wax, scrape a bit away, and then put hydrofluoric acid on it to eat away the glass. It's a nasty thing. It would also eat your bones. So you don't want to be anywhere near it. We're molecularly etching it with DNAs and transposes. They're enzymes. And what they do is they chop up the DNA into little bits. And the bit that's actually protecting them is all of those proteins wrapped around them. So what it will find is the regions where the proteins, these nucleosomes, are pulled away from the DNA, leaving it naked, and then these enzymes can cut it more. And that property of pulling the nucleosomes away to let the transcription factors in is actually the signature of a switch. And so you can do cool things like this. So these peaks are where the switches are. And this is a tiny little bit of your genome. It's only a minor fraction of it. But what actually shows you is where all the switches are. That was a major problem we had to deal with. Look at another cell type, and they've all disappeared again. So you can find things which are on in one cell type and find things which are other in cell type. Cool. So we can sample then the language of the cells. And we do that by using a thing called peak culling, which is where we just use algorithms to go along and pick out the different regions. In fact, we use machine learning for it, and the guy who wrote it is sitting in the back, called Lance. And actually, it's a very effective way of understanding these patterns and finding the bits of DNA where the code is, because it's 3 billion base pairs, remember, and it's moving all the time. We then hot encode this as a standard way of encoding this thing, just a different digit for a different letter. And the good thing about the genome is you have tens of thousands of data points per cell type and across many thousand cell types. So it's perfect for neural networks. So this is very old. This is just a straightforward convolutional neural network from about 5 years ago. And at the top, I'm showing you the real data, and then that's the machine learning predicting it. It does a pretty decent job. And actually, we're getting better and better at this using neural architectures. So what? Cool trick. This is probably the reason, or the underlying system why, I'm going to be melodramatic here, so excuse me, everybody in this room is likely to die, including me. It wasn't a threat. And the reason is when we sequenced the human genome, we were going to find all the causes of common diseases. That's what we thought we were going to find. So we did big studies where we compared people who got a disease and people who didn't get a disease. And we looked at their sequences, and we looked for the little changes that actually make us the unique human beings we are. And to try to find if there was statistical association between having these changes and getting this disease. And they do. And people spent about 20 years absolutely murdering this thing, doing about every disease you can think of, every condition, height, whether or not you can smell asparagus in your urine is in there as well, honestly. The problem with it is they're nowhere near genes. All of these signals are away from the genes. They're actually out in the switches. And that's where we came in. And so the big problem with this is you don't inherit one little change. You inherit a big block of them. It's just the way your DNA likes to recombine when you're made. And so the first thing is you need to know which one's doing it, if any of them are doing it, and how they're doing it. And the other problem, it doesn't tell you what cell type it's working in. You have complex diseases like Alzheimer's. Which cell type in your brain is it actually working in? Very hard to know. The changes may lie in enhancers when you actually intersect them across all the cell types, but they may actually not do anything in that enhancer. There's lots of spaces between those words to actually change things. And you need to know which one of these variants is doing it and what cell type it's doing it in to actually solve it. And to solve it, to understand the biology of the disease and ultimately design new treatments. And the problem is we have millions of these things across thousands of cell types and thousands of enhancers. So that's really emphasizes why we need to have these computational, powerful approaches to decode this. Let me show you some regulatory variation, as we call it. This is a region. There's a gene. That bit at the start is the promoter of the gene where the big molecular machinery lands. These two things on the right are switches. That's a normal human being. And we can do other assays to say what type of things they are. This thing at the bottom just shows that this chemical marker on here shows these are just very active. But then there's donor A. Completely killed these two switches. And there will be people in this room who are donor A and donor B. These are normal people. And this is happening all over your genome in all of the different cell types. And that's why we are who we are, different people. These things are not linked to each other. You inherit them independently. So here's what a neural network can make of this. So what we really want to do is give it a big haplotype, give it a load of cell types so it's learned to code from it, give it that mixture of changes and go which cell type and which change. And what you can do is this molecular etching, and what I've done it from two individuals, and this individual has this change, the red one, and actually has affected this SNP. This shows just the activity of it, but this is actually the machine learning neural network rolling across this, actually predicting for that cell type what is active. And the important thing is to keep your eye here, because if you zoom in, it can tell that that person has it and the other person doesn't. So this is the way you can actually decode those millions of base pairs. So the neural network identifies both the variant and the cell type from sequence. And you only need, actually, important point, which I completely missed, you only need to do this in one person once. So the other problem is how to work out which enhancers or switches are turning on which genes. And there's a very simple thing behind this. There's no quantum physics involved in this. These things actually touch each other in that massively complex landscape that I showed you. And we can use this. We have developed assays, my group as well, have developed assays that can actually capture that event in the nucleus en masse and actually work out which bit of the genome is talking to each other. This is an example of it. This is a thing called tidal C or high C. It doesn't really matter. How you interpret it, this is just a big bit of your genome here. What these triangles mean is this bit is talking to itself much more than anything else. That's all you need to know about it. That's really it is. So your switches and your genes should be in there because they're talking to each other. All there is to it. We would like to be able to predict that because the experiments that you need to do this are ferociously expensive and cannot be done in every cell type. Particularly, you need lots of cells to do this. This is a neural network, which used the first neural network to predict where things are. And then we use a process called transfer learning to then teach it about these, because these signals are the emergent properties of where all those switches and genes and things were. So we can now actually predict from sequence which things are talking to each other. Problem is, this is too low a resolution. These are tiny little things. They're only a few little 100 base pairs, which is an absolutely infinitesimal amount of your genome. But the technologies are getting better. So this is this high C thing. This is your globin lockets where you actually make your hemoglobin. The little green triangles are where the switches are. So this is a run-through of different technologies, improving as they go, trying to work out what is talking to these two genes. The first one here is promoter capture high C. As you can see, it does a terrible job. This is literally a contact frequency plot of what the promoters are actually touching. So high frequency amount touching this bit of genome. It's all over the place. This is very old technology. It's one of the first ones. It's again not very good. This is NG capture C, which actually I developed. And it is pretty good, but I'm horrendously biased, obviously. But you actually compare between cell types, and you can find the things which are dynamically changing their contacts. This is one of our collaborators' methods, and this is astounding. This is single base pair resolution. It can find every single contact in here, and it can do it in thousands of genes across multiple cell types in the same tube. But the problem is, to do machine learning, you need huge amounts of data. But luckily, we just got a nice big grant that's going to let us do this in many, many cell types. So hopefully, we're going to take the low resolution triangle-y thing, get rid of that, replace it with this, and predict at this kind of resolution. So then what we can do is see a change in a person, in their genome, find out which one of them is doing it, which cell type, how it's then going to actually change which it's talking to in terms of the gene. You need to feed the machine. And the problem is, we don't even know how many cell types we've got in our body. But these methods, which Shankar actually talked about, are called single cell methods. And you can literally take a chunk of human, usually post-mortem, and actually pull the cells apart and then do that molecular etching on all of the cells simultaneously in a mixture. And then you use machine learning approaches to pull them back out based on their similarities. You can then collapse these green ones and get that nice big track again. So, I mean, a scientist called Bingren literally dumped out another 222 cell types just by doing exactly this. So the machine is going to get fed, hopefully, by every single cell type. Stage of differentiation development is much more complicated to try and understand how we are wired across the whole human body. The problem with it is, it's a huge amount of data. It's an absolutely astounding amount of data. So what you have now is groups like mine, which are basically molecular computational groups, actually employ JavaScript programmers on their staff. Go back 5 years, you would never think that's the case. And we're building platforms where we can actually ask questions, visualize, and talk to these things. The problem is that's not good enough. I mean, this isn't the full solution. We have millions of these things. These things control genes. The genes have functions. The genes talk to each other. So what I can see, and I really like John McClune's talk earlier on, is why don't we start building networks to actually learn from this data, not the internet and everything else, learn from this data so the scientists can actually, one, you saw that lovely thing where he's actually writing code through language to ask complex questions. Could you do this analysis for me? Not hours sitting coding like I used to do. And then the other thing is actually ask questions of the whole data set is one thing. I think that's where we have to go. The one thing we have to stop doing is stop it being such a bloody liar. Because for a scientific use, it has to be accurate, reproducible, responsible, and backed up time and time again. If you ever play with ChatGPT, it's a great liar. It can sell you a used car anytime. So the take-home genome is big, complex, important for all of us, and in my view, beautiful. It really is what makes us. I mean, why we get ill and die, and how tall we are and color of our eyes, all affects our life and makes us who we are. But the complexity really has surpassed the capacity of that. So we need help. And this is the scientists who did it. Thank you very much.
Thank you, Jim. Very, very nice talk. And is there any questions? Go with Hal first.
Trillions of different cells.
Sorry.
Trillions of different cells, Jim. But if you look closely enough, each of those cells will be different from the one next to it.
Yep.
So how many cell types do you think we need to know about in what level of detail before we can make better progress?
That's a rotten question. Thanks, Al. I mean, there are many layers to that answer. If you think about, sort of organogenesis or embryogenesis that Shankar was talking about, we need to know each of those single cells. And of course, the cells he was talking about that move from there to there, they will have a different landscape by the time they've got there. They've switched on other things because they're talking to different things. The complexity is astounding. Plus, just differentiation of a cell. I mean, we do it in the lab. You can take some progenitor cells from somebody's arm and make them into red blood cells in the lab. And the numbers of stages between that and getting a red blood cell is huge. So the complexity just goes out and out and out. But we get broad strokes. We understand immune cells, which of course you're interested in. We understand blood cells. We start to understand the brain. All of these big broad strokes, we start to understand organs. But there's always more to find out. So thanks for that one.
You mentioned that genome-wide association studies hadn't got very far. What about enhancers?
How far have they got?
Yes.
If you mean in terms of genome
I'm talking about things like schizophrenia.
No, I mean those things have not gone far. I would say that their outcome was disappointing for the people who did them, because we did originally think. I mean, I was a big enthusiast. Thought we were going to find genes. You can interpret a gene. That gene does this. You get an enhancer. It's like, well, there's a whole layer of other stuff that you need to do. And we needed another 15 years of technology to deal that question. That's what we're showing you here, is once we can link that up with a gene, we can get to the point we wanted to be when we started off, which actually give me the gene in the first place and I can understand its biology. So GWAS didn't not do anything. It just took us another 15 years to be able to decode it.
Okay, on that, in the interest of time, I mean, all the speakers will be around for a bit longer afterwards, so I'm sure there's some more questions to answer. Thank you very much, Jim.